Why is a good drug so hard to find? It's because the human body is so enormously complex. Although drugs are designed to interact with specific molecular targets, biologic systems are in fact composed of highly complex adaptive networks that are themselves embedded in other networks. The connectivity relationships among these elements are exquisitely controlled, but difficult to predict. This complexity has presented a great challenge in the increasingly costly process of drug R&D. Until now, it has not been possible to predict or monitor the consequences of molecular interventions on the causal pathways that drive diseases or to navigate toward desired outcomes. The result has been worsening attrition rates of drug candidates, especially when hitting new targets. Most drug candidates fall by the wayside due to lack of efficacy or unforeseen toxicities. Dramatic advances in chemistry and molecular biology have not been translated into new drugs or improved human health. There is a solution. For drug R&D, the missing link is tools for real-time navigation through the critical path to drug approval. The outdated tools for navigating from molecular targets to clinical outcomes need to catch up to the tools in molecular pharmacology. We cannot continue to drive blind. This is where KineMed's platform and tools can make a radical difference in drug discovery and development. Here at KineMed, we have invented and validated a platform of tools for measuring the dynamic activity of causal pathways of disease in humans as well as animal models. These pathways include processes in the brain, such as the transport of cargo through axons in Parkinson's and Alzheimer's disease, cell division, differentiation and death, such as for cancer and blood cells, the assembly and removal rates of key proteins, such as collagen, myosin, and amyloid beta, the synthesis of lipids in blood, liver, and fat, the flow of cholesterol out of tissues, through the blood, and out of the body, the turnover of skin, and systemic processes such as inflammation, coagulation, and insulin action, as well as many others. The activities of these pathways are measured by novel proprietary labeling and mass spectrometric techniques that we have developed here over the past decade. These tools have uses at every step in drug discovery and development: picking targets, choosing the best compound in class, establishing therapeutic proof of concept in humans, identifying the right patients, finding best dose and regimen, establishing variability and statistical power in patients, anticipating toxicities, personalization of therapy, deciding whether to get out early. To learn more, visit our website